So good afternoon. In today's class, we will discuss mostly about the end semester questions. Until now, we have learned, until the third unit, we have learned several techniques. I think I have covered I covered all the syllabus. If there is anything left over, please bring it to my notice. But to my understanding, we covered all parts of it. So if you remember that the title of the paper is Recombinant DNA Technology in Medicine. So the applications are towards the medicine part of it. So we should also we should think of the pattern. I think I already described to you. It is called pattern 4, where you will have six in two. It has part A and part B. In part A, you have 6 questions, out of which you are supposed to answer 4. And then part B has 1 question and you have to answer it. Each of these, so in part A has 80 marks and part B has 20, total of 100 marks. Each of the questions will have sub questions, may have sub questions. So it's not that one essay type. The first thing you have to keep in mind is it is a paper not to be written as an essay. Instead, you are supposed to present something details, that is why I want to discuss about what to expect, what I expect, or how you will be evaluated. I think many are absent, so I hope you will be able to share whatever information that we are discussing now with your other friends as well. So I hope you can see my screen. Can you see my screen now? So the essay questions will be typically something like this. A scenario will be given, which is usually about a disease, something like HIV. That's a part of our syllabus. We also had, say, I forgot about it, diabetes, cancer. We don't have it, but we all have a general idea about cancer, so that is also included. Little more discussion today based on that. And maybe you will be given a hypothetical disease, which doesn't have to have a name, but certain symptoms, conditions will be given, or what kind of mutation caused it, and so on. And also we have epigenetic issues like DNA methylation and histone modification. If you would like, you should try to watch the videos and then come back, so that we have 3 more classes, I guess, 3 or 4. We can always have a discussion about any difficult topics you may have. We also have discussed about reprogramming, right? Reprogramming, how to make induced pluripotent stem cells. In the 4th unit, majority of them are applications, or they are research papers in which we use a lot of other techniques that we have learned in the 1st, 2nd, 3rd units. So typically the question could be will be based on a disease or any of the situations like this, right? And then you will be asked to write different situations. How would you diagnose it, right? The medical applications will come in the form of diagnosis, prevention, therapeutics, and so on, gene therapy. We can evaluate the genome. If it is a genetic mutation, we can find out at the level of DNA using southern blotting and sequencing and many other techniques like that. We can also RT-PCR or northern blotting. We also have many other techniques that to see if a gene is expressed or not, and if there is a presence or absence of mRNA as well. So in all the cases, you should also know about, keep in mind about the homozygosity and heterozygosity. sitting, right? That is another issue that we all have to think of as well. And then for prevention, you may have something like vaccination. If you want to vaccinate a child or so, how are the vaccines brought about? And similar other techniques. You can also use kind of gene therapy for many other. A child is predisposed to a particular disease. How can we address it? And also treatment. How if an individual already has the disease, what are the options that you can provide? And then, as I always want, you should always be able to give the limitations. You should provide the limitations of your approach, because no approach is flawless. Everything has some, in different degrees we have several issues, so you should try to describe about those things. And what we expect in your answers is that you should be able to tell the principles of the techniques that you used. If you just say in diagnosis you are going to do it with northern blotting, say for example, or southern blotting, you cannot just say southern blotting and then draw isolate DNA, this thing. That is too trivial. How would you generate a probe? Right? Southern blotting itself has multiple other techniques, other sub techniques, like where did you get the probe from? How did you get the probe? Right? You can choose to perform one whole methodology, something like southern blotting or northern blotting. So there, those intricate details should be mentioned. If you are saying, I will sequence it, if you want to sequence, how will you do it? How would you get this DNA? How would you perform this sequencing? And if you're performing, which sequencing are you using? And in those cases, do you need a probe or do you need a primer? How would you get the primers? And are you end labeling them? How would you end label the primers? Within a technique, in a way, or a methodology. And that those details are also be to should also be discussed. So and that is what is meant by this one. Again, you will make, say, probe if you are making, or if you're making a end labeling or randomly labeling a probe or a primer. How would you do those things? So try to always be elaborate. These, I'm not expecting that you have to describe in words. I am expecting you to describe in flowcharts. Okay, don't write essays. Even if it is a 10-mark question, a question could be addressed in 2 sides of a paper. You can get 10 marks. It depends on the quality and the details that you have included. That they don't have to be, they shouldn't probably. That's the easier and correct way of doing it, is representing it in here, representing the essence of the answer in diagram or some kind of flow chart with all the details. So try to practice that first, and then you will probably be able to save a lot of time, because you can do it in a very presentable and readable way quickly. If you are writing too many things, typically it's difficult for you to follow, difficult for me to follow as well. So I wish we had a classroom course, and if that was a classroom, if we are in physically in the classroom, we would have had a different kind of fun, but and learning. But this is what we can do at this stage, so we will try to do that. So majority of the things will be represented with flowcharts of the techniques. How you take all the prime, the polarity, 5 prime, 3 prime, all those things should be mentioned. And in here, sorry, I also have to, if you are doing modifications of a ORF for a gene or protein, I mean, if you're going to do mutagenesis, there also PCR based mutagenesis, how are you going to do, and all those things should also come in there. And the flow charts, this is what I meant by intricate details, the actual fine details, or that shows the depth of your understanding, preparation, and motivation for this course. So please practice this. This kind of presentation doesn't come in a typical answers in typical papers. You are going to write at the most 3 pages, for a 10-mark question. That means you should have a plan of how you want to um, present the answer. Okay, and then for to show that you're able to interpret the results, like I have done in many of the lectures, if not all, about how in every technique we learned, in majority of the techniques we learned, my focus is about how we interpret it, because that is the fun part of the technique. If you are reading the principle alone, then we are mugging up the technique. And if we are also reading about the interpretation, then this whole things comes together. In the sense that it's a game, right? About deduction. How do you deduce an answer? I would specifically be interested to know about your interpretational skills in trying to do it. So I could have given a problem and then asked you to answer. But I would want you to think of it now. A question is given, a scenario is given, and then you are supposed to think of how you should present a gel diagram or a graph. For example, if you're doing allele specific PCR, how would the graph look like? If you're doing epigenetic, if you're trying to see if it is DNA is methylated or not, if it is heterozygous or homozygous, again, those gels, how would you represent? I hope you remember that we have done some complex problems on that. So by now you should be better than what I'm already asking for in the end semester. You are better than. So the way you present is what is important to me. And graphs like, say, for example, RT-PCR or any other things, maybe not necessarily only gels, you may also have to represent graphs, how it would look like. Then in every aspect, we should always know the limitations and risks. Try to have a critique of your approach, especially if there are some things like treatments and other things that may come up. In those cases, you should be able to, when you propose a way of treatment, then you should be able to describe the limitations and risks that may be involved. Okay, we haven't reached a strategy, or we haven't reached a stage where all of the any of the genetic therapeutics that we may come up with don't have any problem. There are risks associated or limitations, so we should try to address them. So I am now going to stop talking, and I would read a question for you, and I would want you people to take over and speak about it. I hope I see all the names that are usually actually participating and kind of motivated in a way. I see you often, and I hope you will be participating as well. Firstly, before going into that question, I would want to know if you have any questions on this. Do you have any questions about whatever I described about the NSEM question paper types? Any questions or clarifications you may have? Okay, that's fine then. Yeah. Uh, for next classes, if you have any specific areas to be covered, please, I have been asking you several times, please let me know so that we can prepare on those as well. Now the question comes, I want you people to discuss and let me know what do you think. Is it legible? DMD? Is a debilitating and progressive genetic disease of muscle loss. It is a recessive X-linked disease. the females are unaffected and could be carriers. You are supposed to screen about 100 females who have reported incidences of DMD in their family history. Now, usually I would describe any kind of disease, even if it is hypothetical. You will be given some information on it so that you are not actually, you don't have to be an expert on that particular disease. Okay, some information will be given to aid you so that you can pick up from there. Um, Describe a method to diagnose if an individual is a carrier or not. Draw representative schematic gels to differentiate homozygous and heterozygous individuals. That's the first thing. Second question B, question B is B part. Describe the intricate details about the methodology and techniques that could be used to treat a child using gene therapy. C is describe the options available for a carrier female who wants to ensure to have children without the mutated dystrophin gene. So these are three questions in total. They may be 5 marks for A, for B 10 marks probably, and C 5 marks, something like that. A total of 20 marks will be given on that. Expected you to watch the video on DMD. I hope you have done it by now. Even if you didn't, you should still know how this problem could be approached. There is no one correct answer. It is an open-ended question. At least you can come up with other valid or qualified strategies too. What we discussed in the video necessarily doesn't have to be the only answer. All I need is that you should have taken or identified proper approach or designed a proper approach or strategy, and then picked relevant techniques from various in addressing the scenario. Okay, questions will be specific like this, and some particular answer that you might have to come up with, and that you should be able to do. Okay, I will now mute myself, and I would want to see what you are going to discuss. I hope it will be useful. Please participate, and each other should be able to critique on others, and please present your views on this question. Okay, who is going to start? Ramanand was first in the class in this classroom today, so he will start. So that I just want to watch where we are. I'll have a good gauge about. I'll be able to gauge where we are, and we'll try to use the other classes for any other purposes directed from here. Okay, please somebody start, and let's discuss. Yes, you can take a few minutes and then no, come up with take 5 minutes or 10 minutes and then come up with a strategy, overall strategy first, and then we can discuss. Has anyone come up with any solution so far? If you did, please, if you have it on paper, you can present as well. Is anyone ready, please? We are okay. Can you describe it so it will be open for discussion, Akshaya?
Yes, sir.
Yeah, can you describe so that we can have a discussion? That's the objective of it. Okay.
Okay. Yes,
Yeah.
sir.
Right.
So for method of diagnosis, you'll have to do DNA testing. So the carrier female might not have the gene which produces dystrophin, and that'll be gene XP21. Um, the If you want to see physical features in the carriers with symptoms, then that would be protrusion of the belly because of bending of the back, and swollen calves and hands which are backwards to balance the weight.
No, no.
Uh,
Sorry, one second.
yes, sir.
Sorry to interrupt. So when you say diagnosis, how would we do that?
So you'll test the concentration of creatine kinase in the blood.
Here we are talking only about carriers. Carriers are they are the females and carriers. That means they do not have other symptoms.
Yes, sir.
Okay, so that is, if it is affected person already, if it is mostly it's evident in the boys,
Hmm.
males, that then it is easy to find out physically and other blood tests. But here
Okay.
we have to go, because they are carriers, we have to go for a genetic diagnosis. In that ways, how would we do those, right?
Okay, sir.
Yes, yes, yeah. Anybody has so see, I don't want to speak, others should speak as well, and then that is how we all can grow together. Please, it is about critiquing each other, and the objective is to better each of us, our understanding, and the answer itself. Okay.
Okay,
Yeah,
sir.
yeah, continue. So sorry, so I hope you all are here listening carefully. The questions will have specific details, and the answer should then therefore be highly specific to the given scenario. General answers will not be may not be correct or appropriate enough. The depth will be lost if the answer is not highly specific. Okay, so try to think of about the scenario and then try to address this. But yeah, full marks to Akshaya for actually putting up the answer. Okay, that's what we all should, I think. When I was trying to write something somewhere, we need to, knowledge has three things. One is intent, why are we seeking that, right? Second is content, what are we seeking? And the third one is the most important one, audacity. We should have courage in all the aspects about having an intent, to courageous to have an intent, and courageous enough to seek the knowledge and acquire the knowledge. And the third one is courageous to communicate the knowledge. If we are shy, or if we feel, or if we are afraid that somebody else is going to judge us based on what we are saying, then we are not worthy of the knowledge. So we have to practice this audacity or courage to in all areas of desiring, deriving, and communicating the knowledge. So try to put forward your views first. Even if they're wrong, that's all right. We are all close friends here, say. We don't disrespect anybody here, right? And that is the place, when there is no judgment or when there is no disrespect, that is the place where knowledge comes about. So again, I'm asking everyone to please put forward your views. There is nothing wrong in that, and it's always a good thing to put forward views. That is when we will learn how to put forward, how to better put forward our views in a better way, right? We all should be tested. Yes, continue, Akshaya. Thanks for the posts.
So I was focusing only on the creatine kinase. I'll have to look up a bit more about the dystrophin gene. But
Okay.
if you're
Yeah.
going to do a gel, then the heterozygous will be above the homozygous uh, homozygous gel.
No, yeah, we have to draw the picture, otherwise
Yes,
it's
sir.
difficult to describe in words.
Okay.
Yes, that's right. Can anybody tell me what diagnostic tools can be used in this case? Where in DMD, majority of it is because of a mutation, right? It could be a small mutation, like in the last slide. I'm not sure if you all watched the video or not, but it is deletion. There is another dystrophy which is a large deletion, and there is another, the DMD is a smaller deletion. And in DMD there is also a problem of frame shift mutation. So if we know about the mutation, no, I shouldn't be speaking. It's about you people speaking. Yeah, try out, and can you please suggest some different techniques that could be used to do to find out these things? Anybody?
So are the symptoms different in a person with a frame shift mutation and with the deletion of the entire region? Because
Uh,
in the video there was a stop codon which occurs in the middle, so it's only a part of the gene which goes in the frame
yes,
shift mutation.
that's right. That's right. Yeah, I mean,
So an electrophoresis will be enough to test whether the person is a carrier.
how would you get? What would you electrophorose?
Oh, okay.
Anybody?
Sir, I have some idea, but I'm not sure if
Yes.
it is.
Don't be sure about it. Just say whatever you comes. Don't worry about anything. Yeah.
We can amplify the gene sequence, not exactly the gene which we are looking for,
Hmm.
somewhere around it also, let's say 500 base pair long. So
Okay.
we'll amplify it on both the chromosome, um, and then we can use a restriction enzyme to cut in between the gene, uh, the required gene which we are looking for, and if
Okay.
it generates uh, shorter bands, like two shorter bands, means
Yes.
the particular gene is present, and if it doesn't, means whole length 500 base pair will be there. So like if it cuts on both the chromosome, we will have only two shorter bands, and if it cuts only on one chromosome, or the first one and not in the second one, then
Mm.
we'll have one full 500 base pair and one like two small ones. So we can
Okay.
tell that if
Yes.
so
What you said
only one
is,
X will have.
yeah, sorry.
Yeah, right.
Yeah, what you said is correct, provided if the mutation is going to affect the restriction site. Is that true?
Yes, sir. That's what. It's not necessary that restriction site will be there also or not.
Yes, that is also another limitation. Yes. So,
Yes.
what do you suggest? What else can be done?
Um, I'm thinking. We'll see.
Yeah, there are actually multiple techniques one can use. It's not just one technique. You have to think. Don't narrow down your approach in the beginning itself. Think of multiple possibilities. If there are more than one technique for this one, and that is why it is open ended. All it your the technique that you choose makes it qualified if you are able to justify, and if the technique is applicable to the given situations. Okay, yeah, keep
Yes.
trying. Yes, if anybody else also has.
Sir,
Yes, please.
sir, DMD is like the gene is getting deleted, so we have the disease coming into picture, sir. So, why can't like karyotyping be done? Because if we identify that if that gene is deleted, so we can confirm that the person has the disorder or has the deleted gene in that. So, why can't karyotype typing be a tool for that?
I didn't say it cannot be used. You defend how it can be used. It
S-
can be
uh,
used. There is nothing wrong with it. Yeah.
like if as Divyanshi said, if we have to amplify, use restriction enzymes and all, it might be time consuming, but for karyotyping we can take that blood and we can like arrest the cells in the metaphase stage, and then we can analyze the chromosome.
Yeah. How long does it take to do karyotyping?
um,
I think you have to grow the cells over for 2 days or something, I guess. One day
yes
at
sir.
least.
Yes
That is
sir.
going to take, just growth itself is taking one day. What we should, if we want to test for, if we want to, in karyotyping, we are going to see the band patterns and sizes of the chromosomes. If the mutation in DMD, in dystrophin gene, for example, is not going to affect, if it is not, okay, hypothetically,
Okay
if
sir.
it doesn't affect the banding pattern, because if the mutation is very small scale mutation, then you won't be able to see it on the karyotype. The mutation, if it is a large part of mutation, then yes, you can see it.
Okay sir.
Yeah, that is what you should consider before doing. Not that it shouldn't be done. I think it is done also in some cases, at least.
Okay.
Yes.
So
And
can
actually,
Western blotting? Sorry.
karyotyping is little long. It takes more time than the some of the molecular techniques. Yes. Yeah, you continue, please.
Yes, so like I just had a rough idea of since it's deletion, why can't we detect it using karyotyping, sir? But
Okay,
yeah,
okay. Yeah, that's fine. That's fine. That is also a correct answer in a way. Yes.
okay, sir.
So,
Anybody else? Yeah, Akshaya, continue.
so can Western blotting be done using the blood samples for the presence of the dystrophin protein? We
Why would
can.
you expect dystrophin in blood?
Okay. Tissue samples?
Yeah, you should have taken skeletal muscle where
Yes, sir.
dystrophin is usually expressed in large quantities. Then you should take a biopsy from the muscle,
Yes, sir.
and
Sir,
then yes.
sir, if the patient is if the person is a carrier, why will she not have the dystrophin protein? She will have it, right?
No, they see it depends on expression of both. If the person is going to have, if it is heterozygous and both are expressed, she may not, the muscle is not dying, but there is a probability of the truncated dystrophin also expressed.
Okay. Okay.
Yeah, I hope I made a point.
Okay,
In
so even if she is a carrier, she will show indications of
I'm not sure about it, but there is a probability that she might, that gene also is expressed. Maybe they are degraded faster, or many some other thing might be there. In men, if it, because it's X-linked, there is only one copy, and if it is a mutated one, then it is a problem. In females, you have two X chromosomes. Even if one copy is bad, the other copy is going to supplement or kind of complement the loss, and
okay,
that's
sir.
how the females are asymptomatic.
So they have milder symptoms. They're not completely asymptomatic.
Yes, yes, probably, yeah. That then, if then it would mean that the truncated one is present.
Yes,
Yes.
sir. So instead of a complete karyotype, we could do a PCR itself of just that region.
Yeah. Say what you want to do.
Uh, so we'll have to know the gene sequence and use primers of the regions before and after the
Yes.
XP21, and through PCR we'll be able to detect the size, and
Yes.
if the gene is smaller, then we'll know the person is just a carrier.
Yes.
Sorry, yes, sir.
If
If it's smaller, it's a carrier. If the gene is present, then the person is
I
not
mean,
gonna
if
have
it
DMD.
is homozygous normal, you'll have one type,
Yes,
one size band.
yes, sir.
If it is heterozygous or a carrier, you will see two
You'll get
bands.
two
If it
bands.
is
Yes, sir.
based on the deletion, you can
Yes.
also do, if we have complete knowledge about the mutation, Then we can also do allele specific PCR,
Yes,
where
sir.
we design where the ultimate 3' end base is, it could be pairing or not pairing. I hope you remember we discussed this in one of the videos. You please have a look at those. There could be, you could actually do southern blotting also if it is a large enough deletion, right? You can do you can probably do western blotting if you're, as you're saying, if we have if we have antibodies towards the amino terminal of dystrophin, which is present in full length as well as truncated ones. If you see two different bands in western blotting, that means there is a full length one and there is a truncated one, isn't it?
Yes,
Otherwise
sir.
dystrophin should have given only one band.
Yes, sir.
So there are it's all about how you use and if you understood the scenario that is given clearly or not. So these are the things one has to keep in mind when you are addressing the situation. Okay, it's not about we telling what we know, it's about we telling what is asked for. So that is when the answer becomes little critical, or it's kind of highly specific. We will quickly go through B and C because we are running out of time. So B is describe the intricate details about the methodology and techniques that could be used to treat a child using gene therapy. This we have done according to the paper that we have read. Gene therapy in general can be done in vivo or ex vivo. In vivo is you're directly giving the gene to the body and hopefully the target cells will take it up and cure the person or provide a functional copy of it. In that case, if one is trying to do that, um, what are the methods that should be taken? You should have cloned the gene, normal gene, then you should have put it into an appropriate vector, then you should have put it into vesicles or so that could target, specifically target the cells we intend to modify, something like skeletal muscle cells in this case. That's in vitro, in vivo gene therapy. If it is ex vivo gene therapy, we should have taken the cells, some differentiated cells, made induced pluripotent stem cells, then corrected the gene in that particular cell, and then put back, again derive the differentiated cells of muscle progenitors or so, and then put the muscle progenitors in different parts of the skeletal muscle cells in the body, which could probably take over and try to correct the defect. That is what we learned in the ex vivo gene therapy is what we learned in the last lecture. Okay, those are at least some of the ways how you would go about, how would you clone the gene, and all those details should come in when you're trying to describe an answer for this. Okay, how would you make induced pluripotent stem cells? You should have also described about it, right? And for that we have used another paper in which we studied piggyback vectors or you could have used retroviral vectors to use the pluripotent factors or Yamanaka factors, OSKM. So, and then uh, made the pluripotency. And then using PAX3 and others, you could again differentiate into progenitors of stem cells, skeletal muscle myocytes, and then after modification, of course, after gene therapy, gene correction, and then putting into the body part and the muscles. That would be the answer for the details about how we do treat a child who is suffering from DMD. Then we have the third one is I asked is describe the options available for a carrier female who wants to ensure to have children without the mutated dystrophin gene. If one wants to do this, there are two options, right? One is to go for germline therapy, take an egg cell or make the correction in the egg, and then allow fertilization to happen, and then put it in vitro fertilization. I mean, in vitro fertilization, then transplantation, and the baby is born with without the mutated gene, or with two copies of the normal gene, or one copy of the normal gene, as the case
So,
is required. Yes, please.
sir, can't we directly select an egg which does not have, because it's either this X or that X? Can't we
I
select
know.
the X which does not have the
That's what
mutation?
I was about to say, the second one. Okay. First
Okay.
one is germline therapy. Trying to, you can take Hugo stem cells. I mean, that in humans may not be, it's not be possible. But there are techniques coming up where we can generate gametes from normal cells also. In that case, we make the correction and then get the gametes from female, and then put it into normal, allow fertilization to occur, and so on. That is germline side. The other one is you allow fertilization to happen with multiple eggs of from the female. Then you do prenatal diagnosis to see if any of the males, if any of the male embryos have the mutated gene, and eliminate from the population. So you would actually select those male embryos which have the proper or the normal X chromosome, and then you put it into. Is that what you meant, Pratana?
Yes, sir. Yes.
Yes, that those are two approaches. You have to talk about both. That's why the question asked, describe the options available. That means it's not one option. You have to think about multiple options, and then try to address how one can have a biological child. Okay, now you cannot say maybe they can adopt a normal person, and that's not an answer one would expect at this stage, isn't it? So uh, so it's all about how deep you think. You will spend more time thinking rather than writing. It's an unlike many other subjects probably you have done, you have written so far, biology ones especially. We keep writing for 3 hours. That's not what is going to happen in a typical RDNA exam. You are supposed to spend more time thinking, and come up with very appropriate and specific answers to the question, and try to present it in a as a flow chart, and with the critical notes here and there. There will not be any essays in this whole of the paper, nowhere. Yes. And I didn't add, there will be a question usually that will come, which will ask you for what are the limitations and risks involved also. That is another problem we'll have. So if we are using in vivo gene therapy, what if the non-target cells are affected? Then it's going to be a problem. And then the integration of the cells, the vectors might cause mutations and cancer maybe. We don't know about those things. So that we also don't know if it is ex vivo. We don't know if the incorporated cells, those cells that we have put in, we don't know how long they will survive. Many other things, how fast they can integrate and stop. These are some of the limitations we have to think about when we are taking any of these therapeutic approaches. Okay, I think the time is up. We will discuss. Please let me know if there are any topics that we should have discussed. Then I will be able to make a proper presentation or class for the next time. Yes. Okay, take care and have fun. We'll see you in the next class. Bye bye.